I'm not going to talk for very long, in fact, because I'm not an expert on nanotechnology. My field of interest is viruses, which are nano, of course. Natural nano, should we say. I'm going to call them natural nano in future. Sounds much more impressive than viruses. And immunity. And so why am I involved in this at all? Well, because I'm very senior and obviously right on the way out in science, and it's very interesting to interact with broad and very dynamic areas of science and to do what you can to help things to go well. So I was asked if I'd be chair of the Australia Research Council Centre of Excellence in Nanotechnology and Biomedicine bid for funding, and the funding got up, and so now I have to do the job of being chair of this thing. And I know I'm going to learn a lot from it, and I sat through a lot of the session this morning, and I've already learnt quite a lot, which was really quite fascinating. Now, this is a public forum. When I look around the room, most of the people have those colourful things around their neck, which would suggest that they were at the meeting. Do we have any members of the general public who weren't involved in the meeting? 1, 2, okay, and good. So we're talking to you. And I'm going to ask, most of this is going to be a question and answer session. We're going to have 4 very eminent and experienced scientists up on the stage. They're going to introduce themselves, and 4 of them, I think, are more on the chemistry side of things, which is what nano's about. And we have a biologist, a physician also, who's in touch with the clinical realities, because that's what a lot of this is about, trying to improve medicine, improve treatment, improve diagnostics, and all sorts of various applications, some of which are really quite fascinating. And the technologies are terrific. For instance, we heard earlier today about a test that detects very, very small amounts of things, but if you've got a lot of it, it doesn't work. I think that's absolutely wonderful that you would have a test like that. It appeals to me greatly. And some of these tests are more sensitive than the polymerase chain reaction that we use as the sort of gold standard in virology for detecting whether something is actually there. So I guess I've learned a lot there too. So to head off, well, nanotechnology is, nano is 9, 10 to the minus 9. That's very small. It's 10 to the minus 9 meters. And viruses are very much in that range. The smaller viruses are in the lower end of that, and some of the bigger ones go up a bit. You know, the reason viruses were characterized early on by their size. Before we could actually grow them in tissue cultures, which is way back, you know, in the late 19th century, Pasteur and so forth worked out how to grow bacteria on plates of blood agar or in various sort of soups and things. It was all cookery. I think they got the basic techniques from their wives. In fact, that was true in one case. But viruses couldn't be grown in that way. They had to grow in living cells. So the way they were detected is if you couldn't grow the thing, but it would pass through very small filters, then you called it a virus. And the filters were actually candles of sintered porcelain of various pore sizes. The ceramics industry, and this field is also about material science because that's what ceramics is. The ceramics industry was really quite advanced in the late 19th century. So if they went through those filters, then we called it a virus and we said it's very small because the pore sizes were very small. Now, what that did though is it excluded other things which are actually viruses which are quite big. And recently we found there are actually some quite big viruses by various techniques. They're not important viruses in infectious disease. But anyway, that's the sort of sizes that you're talking about. And they're very tiny things, and they're tiny things that we're going to inject into us or we're going to use in diagnostic techniques and all sorts of things. And you'll hear some of that, and I think you'll hear some of that through the questioning. The Australia Research Council Centre of Excellence is just kicking off now. And it's a type of funding mechanism that we've done very well in Australia. You know, we're a relatively small country in population, as we all realise, and we can put together a relatively small scientific effort, though we do focus that effort into various areas of genuine excellence, I believe. And the other thing we do here that I think we do better than, say, the United States, where I've spent much of my research career, is we pull everyone with talent in a particular area or on a particular problem together from the whole country. And we've done that in the medical area with the National Health and Medical Research Council program grants, where you will have the lead scientists, the chief investigators as they're called in Australia, often in different cities and certainly in very different institutions. And you might think that doesn't work, but it actually works quite well. There's usually a retreat every year where everyone gets together, including the people working in the programs, and the chief investigators get together more often. Now, these ARC centres of excellence are on that model, but they're even bigger, and they're funded for 7 years, and they're led by people who are already pretty eminent, and work as the chief investigators, and then we have associate investigators and so forth. So they really pull in a lot of talent, and there's certain flexibility in that you can, to some extent at least, move people in and out. So this particular centre of excellence involves, I think it's 5 universities and in various parts of the country. Quite a bit in Melbourne, Sydney and so forth, in 4 states in fact. And there are also very good alliances with other institutions and other senior scientists in other countries, both in Europe, United States and also in Asia. Particularly strong is the link with the University of Warwick, where some of the people in the centre actually have joint appointments between the two of them. Now, if you're thinking, well, this is all paid for out of the tax dollar, it's actually, I think, a pretty good expenditure of tax dollars. Firstly, this type of science is very much oriented at trying to get solutions to real problems. And in some cases, creating solutions for problems we didn't know we had, but when you can identify them, you can really understand what's going on. And that's been happening for years in medical science. Everyone's pretty experienced, and I think, from what I've seen of it so far, that it will actually work very well. And if you think of government expenditure, the most thoroughly reviewed and thoroughly assessed and most closely watched money that comes out of our federal government really goes into science through the peer-reviewed funding agencies, the National Health and Medical Research Council and the Australia Research Council. And I think, especially after working for a long time in the United States, we get an extremely good bang for our buck in Australia. And we do a lot with considerably less in real money. So, in nature, of course, many of the interactions that are very important to us occur at the nano scale. Protein-protein interactions regulate many of our cellular processes, and then, of course, there's the nucleic acids and all the rest of it. So a lot of what goes on, the machinery of the cell, is, of course, going on at this nano level. So what are we trying to do here? We're trying to intervene in that in some way, or take advantage of it by introducing entities into the cell that we want to get into there. And we've got a lot of technologies for doing that, many more than I realised. One is to use, there are various types of structures that can be used, for instance. And this biological nano interface is really what we're at. So what does that mean if you're going to do serious science in it? Well, for a start, the chemists are the people who do the nanotechnology, and they have one type of culture and one type of understanding. But if you're going to use it, you have to work with biologists who are different again. So you're interfacing 2 different cultures, and those 2 cultures learn from each other. And that's what we have here. We have biologists, research biologists, we have medical doctors, and we have chemists who are working, say, particularly in this institution and other institutions around town. So the little cartoon, you can see a nano sort of thing going into the cell. This stuff is interiorized by various ways, usually just by endocytosis or pinocytosis, goes into the endosomes. Though there are also, we also heard about needles that actually poke in that looked very impressive. A nice little video, and you can see these needles going into the cell, but they don't go through the nuclear membrane, which is extremely interesting. So these structures can be circular, they can be tubular, they can be needle-shaped. For nano, they just have to be very small. And what are you using them for? You're using various substrates to actually, in many cases, to actually carry things into the cell to get them to various sites. Now, one of the big problems in say, I've gone too quickly, in say cancer therapy is that the drugs that we use to treat cancer are basically poisons. What they do is they poison the cells. So what we've done traditionally is we've got a cancer patient and we load them up with some toxic drug. It has horrible side effects. Their hair all falls out, their bone marrow may be destroyed, and it's truly awful. So there's been a long-term effort in medicine and in our thinking, which hasn't often gone all that far, to try and target these drugs precisely. If we can actually target that drug to the cell where it's needed, then we can do an infinitely better job. We can get it there in much higher concentrations, and we don't have all those horrible side effects. So what you can have on these nanoparticles is various molecules on the cell surface, or structures on cell surface, which will actually target them to this cell or that, or if it does get into the wrong cell, will only be activated, say, in the cancer cell. So there are all sorts of neat tricks in this, and of course those of you who've been sitting through the meeting, and of course many of you work in this area, will be much more familiar with it than I am. I also heard for the first time the term therognostics, which is wonderful. What you can do is you can engineer these tiny particles so that on the one hand, they will deliver your toxic drug at high concentrations to where you want it, but on the other hand, they will also send out a signal so you can pick it up in magnetic resonance imaging to know that it's gone to the place where you want it. And that's really very neat as well. So you can combine these two functions within the one entity. And it's really, that's reporting, if you like. Really quite fascinating. And there you can see this illustrated. On the one hand, you can have the reporting functions with sensors, imaging technologies, diagnostics. And on the other, you can have delivery. And delivery could be a cytotoxic drug. It could be a vaccine entity. And again, if you can get your vaccine to exactly where you want it, you can use a lot less of the material that you would require to actually get a good immune response. Again, so if you can get it into exactly the right cell, it's a very powerful mechanism. So this is a field with enormous potential. Obviously, we're not going to have it to ourselves. There are going to be very strong players all around the world, and but I think it's something that can successfully be done. Here's an example of a nano drug, a pegylated liposomal form of doxorubicin. Well, that's very nice. And pegylation, I always like that term. I'm not going to explain it to you. The committee, the people on the stage are going to explain pegylation to you. That's one of the first questions, right? And the nano patch, this is a vaccine delivery system that was developed, I think, in Australia. And here we have interfering RNAs, putting them in to actually block a function within the cell, and again using the nanotech particle. Now, the nanotech thing, it takes various forms. As we heard, the small nano-delivering core, if you like, could be a gold particle. It could be something that actually just forms a scaffold when you're making it, and then you put whatever you want to deliver around the outside, and then you dissolve it away. There's all sorts of possibilities, something that stays, something that gets dissolved and goes. Really quite intriguing. And again, here you see an example of a cancer therapy experiment, which I'm not going to go into. So there are issues, of course, and like any new technology, there are concerns in the broader community. It's novel, it's new, it's injecting stuff into people. That's not always injecting stuff into people. Some of this is diagnostic tests that you can use. We also heard an application where you can have a little battery-powered thing which will vaporize from a chip and get deep into your lungs. The nano-sized particles getting deep into your lungs, and maybe the next generation generation of puffer for drug delivery. We'd like to deliver a lot more drug that way. I think it would be a very efficient way of delivering it, and it would be a lot less intrusive. People don't like being injected with things. Where are the issues? Well, like any emerging field, the regulatory mechanisms have still to be worked out, and I'm sure that's an area of great discussion. And then, of course, there's the health and safety issues. And any new technology, especially if it involves putting things into people, will raise those health and safety issues. And you know that there have been all sorts of discussions about the safety with nanoparticles, focusing particularly on sunscreen, I think, which I use a lot of. And if nanotech particles have influenced me badly, I'm not sure yet. So anyway, that's all I'm going to say. You don't have to listen anymore to someone who's talking about a field they don't really know anything about. You know, my role in this has been the role that you find in literature. You know, if you're describing, you know, you read a novel, there are various types of ways of writing that novel. I tried writing a novel, actually. It was so bad it didn't get past my agent. It's one of the worst novels ever written, but never got anywhere. But you can have the informed observer writing the novel. You can have the novel written in the first person. And then you can have an individual called the unreliable narrator. Now, the classic unreliable narrator in a novel that most of us would know is Nick in The Great Gatsby. I mean, he tells you what goes on, but does he really know what goes on? And so I'm the unreliable narrator. And now up on the stage, we are going to get our panel, and they are the reliable narrators. And we're going to ask them questions. Now, this is finishing the session. It's a chance for everyone to ask questions, but I would ask that those who are asking questions and also those who are answering put those questions in accessible English. I used to say that the technique you have to learn is, when you're talking about science, to talk in a way that your grandmother will understand. Until someone said, well, my granny's a nuclear physicist. But so put your question in accessible terms, and as our panel are very distinguished and very experienced, I'm sure they will answer in ways that any normal human being, no matter how geriatric, can understand. So I'll invite the panel to come up and sit on the stage. Yes, give them a hand. Now, with respect to questions, we can address them directly to members of the panel. You can address them through me if you want, but I don't even know enough to know who I should probably ask with the particular question. So what I'm going to do is ask the panelists to go through and just very, very briefly introduce themselves so you know who they are and what they know about.
So my name's Leslie Yo. So I started off at Monash in the Department of Mechanical and Aerospace Engineering. And then 3 years ago, I moved to RMIT in the School of Electrical and Computer Engineering. Now I'm in the School of Civil, Environmental and Chemical Engineering, doing biomedical engineering on biomedical devices. And so what does that make me? Confused, I guess.
My name is Michelle Halls, and my background is in pharmacology and cell biology. So I did my PhD at Monash University in the pharmacology department, and then moved to the University of Cambridge to do a postdoc there, looking at activation of proteins at the single cell level. And I've been back here at the Monash Institute of Pharmaceutical Sciences for a bit over 3 years now, and trying to really understand some of the detail that occurs at the single cell level.
Thanks. My name is Molly Stevens. I'm from London, so I'm also extremely confused. So I did a pharmacy undergrad and then a PhD in single molecule biophysics, just because it sounded hard. And then a postdoc in chemical engineering, and now I'm joined between materials and biomedical engineering. And my group develops different types of materials, some of those which involve nanotechnology for biosensing and also for regenerative medicine.
So my name is Andrew Scott. I'm probably even more confused than all the prior speakers as well. I'm a clinician scientist. I actually did my medical degree at the University of Sydney, quite a while ago now actually, and did my medical training and internal medicine training and nuclear medicine training in Sydney. And then I spent 3 years in the US working at Memorial Sloan Kettering Cancer Centre, which is where I learned about positron emission tomography technology. And I also became deeply interested in antibody and antibody therapeutics, working with Lloyd Old, who was one of the founders of modern tumour immunology. And then I came to Melbourne, south of the border, subsequent to that. And I've been involved with the Ludwig Institute for Cancer Research since that time, and also I see cancer patients at the Austin Hospital, and I established and run the Centre for Positron Emission Tomography there. My research interests are principally in antibody-based therapeutics, receptor-protein interactions, and also in imaging, particularly in cancer, using multiforms in animals and humans.
So I'm Angus Johnston, and I sort of continue the theme of people who have gradually shifted fields. So I did my PhD at the University of Queensland, ostensibly in physical chemistry, but trying to understand sort of biological interactions in a physical chemistry way. And so as my career has developed, I've sort of gone more and more down the biological path, because as we tried to develop materials for drug delivery, we found that our understanding of what was happening was very limited, and so we needed to sort of go back and understand the fundamental interactions of these materials before we could actually know what to design for the perfect delivery system.
So there you have a little bit of an overview of modern science and the way it's going, because what's happening is people with all sorts of different expertise are coming together to look at real problems. And so that's why a research university, for instance, is such a powerful institution, because we have all sorts of different technologies within the institution. It's very broad, and that's why it's so powerful also to have these interactive networks. And as many people said, they started out to do one thing and they ended up, or they're doing something else. They're certainly not ended up in any sense, and they're a bit confused. Well, I've been confused forever, and I'm still trying to decide what I'm going to do when I grow up, but that's a bit the way science is. We're constantly sort of probing, and what makes it wonderful is when you can talk to great colleagues who know totally different sorts of stuff, and you can bring those two things together, and that's where a lot of the power of science goes. Okay, so now we're going to open it up for discussion, and do we have some questions? Because if we don't, we can all go home. I mean, but I think they've taken the booze away, so questions? Yes?
So one of the big challenges in cancer is the fact that we have to wait so long for people to get their secondary cancer, and then we treat them. And secondary cancer is what causes 90% of cancer deaths. The reality being, probably by the time most people are diagnosed, the cancer cells have already seeded. How do you think nanotechnology might help us to actually go after those isolated cancer cells, the seeds of cancer cells which might be hiding in the bone marrow or somewhere else, and get at them early? And so the challenge you have is not only are you trying to get at them early, but you're actually treating patients who are healthy. They're not patients with advanced cancer. How are you going to get at them and take drugs to those sites and have the same sort of therapeutic index, if you like, that would be tolerated in someone who's basically a healthy person?
Now, I know Molly can kick this off from the point of view of diagnostics.
Yeah. So I can kick off, and then maybe I think everyone can probably contribute a little bit on this one. So
it's a really good question.
So some of the ways I think nanotechnology could help is in making new diagnostic tests that could just be more sensitive than the sort of tests that are available now. So, for example, if you've had a cancer and you've had it treated and you're looking for recurrence, so for example, like the prostate cancer, if you'd had a surgery, had it removed, and you're looking for very low markers of disease, so tiny amounts of cancer that might be recurring, then making tests that are just more sensitive are going to be able to pick that up earlier. And that's the sort of thing that many groups are trying to do, including our own group, but many other groups as well. So I think getting that much earlier diagnosis is key, really, for diseases like cancer, to be able to then get the better treatment. But other people can talk also about imaging and how the drug delivery might kick in.
Yes, I guess I'd touch on two areas. The first is I agree that being able to develop various types of biomarker-based tests, in vitro tests, is incredibly important. And there are many, many platforms, as you said, that are being explored, both from blood, from urine, and many different sources. And at the moment, in terms of clinical development or clinical trials, the ones which are probably looking quite interesting at this stage are more exosomal-based tests, circulating tumour DNA. It's fairly routine now in clinical trials to be looking at detection of circulating cancer cells. The advantage of those approaches is not only can it detect whether cancer may be there, it may also give you indications as to whether there's specific mutations that are there or changing, which can obviously guide therapy. And so there's a lot of, I think, in vitro-based diagnostics which many of the big pharmaceutical companies as well as regulatory agencies are looking for very actively at this stage. From an imaging perspective, which I also am involved with quite a lot, it all boils down to resolution and specificity. So we know that some of the more high-resolution CT-MR-based approaches are extremely good, but always the question of what's normal versus abnormal comes up. In the context of PET, and I declare my bias here, PET obviously allowing you to look at a broad range of metabolic processes which may or may not be specific for cancer. You can really pick up things down to the 3 to 4 millimetre size within patients. But I think the other important thing is that either with MR or PET-based probes linked to nanotechnology, that may be another way we can be more specific about detecting where residual microscopic clusters may be. So you do a diagnostic in vitro test, and then you can image to see where it is and decide, is this something that would be best treated locally with radiotherapy or surgery, or should you be giving a more systemic-based treatment, and what sort of treatment would you give? And going to Peter's comment before about theranostics, which is the big buzzword at the moment, if you can develop something which can help with your imaging and then be used for therapy, for payload delivery, for whatever, that's a very exciting area. And from a regulatory perspective is also very good, because new therapies for cancer now are very expensive. And what regulatory agencies want is a better way we can predict which patients will respond and won't. And theranostics is a way that you could potentially do that.
And I suppose I can say something on terms of the delivery aspect of things, and I think it might have been something that when we first doing some work together, Andrew said, was that the goal for our drug delivery isn't to try and treat the primary tumours, because that's removed through surgery. It's all the metastases that we're really trying to go after, and they're the ones that you can't see, and there may be hundreds of them going around. And so where the nanotechnology can really help is that you still will have the surgery to remove the primary tumour, but then rather than having the chemotherapy, which is at the moment pretty much a scattergun approach and hitting all the healthy cells as well, if we can then clean up those remaining metastases with the targeted delivery, that's where I think some of the real power comes in for the nanotechnology. There's obviously the major challenges of actually keeping that specificity once we go in vivo and make sure that we don't have accumulation in healthy organs, and I think that's a real challenge that we have to meet.
Do you want to go? So I work in a field called microfluidics, which is pretty much like small-scale plumbing, and the goal of this is to create devices that people can take and use to do cancer detection, for example. And I was talking this morning about the concept of a whole lab on a chip, where it's like Honey, I Shrunk the kids, but I shrunk the lab instead onto a chip scale device that you can go out and hopefully buy from your pharmacy, just like a pregnancy test kit for $5, and hopefully it'll detect, it'll tell you if you have cancer. So the holy grail in the whole field right now is to do cancer detection. There's a lot of talk about trying to detect circulating tumor cells for early stage detection. The problem is that it's like looking for a needle in a haystack. I mean, you've got this tumor cell amongst, billions of other cells as well. And so I think the field is shifting in some ways, that people are realizing that that's not so easy. You can take blood out and you can try to screen cells for the mechanical properties, for example, and tumor cells would have a different mechanical or stiffer membrane, and so you can trap them. But I think people are realizing that it's not so easy, and so as Andrew alluded to, we're looking at different ways now. For example, there's a lot more DNA, circulating tumor DNA or exosomes or microRNA, where cells signal to each other, and they do that by releasing exosomes. So that's all happening. Whether we have devices that can do it sensitive enough, in the point of care, that's another story for something to look at within the next 5 to 10 years.
I think also sort of expanding on what Angus was saying, that there's a huge potential for this, I think probably a little way down the track as well, not just targeting the tumour cells, but being able to target subcellular areas of these cells too, because a lot of receptors are highly overexpressed in tumour cells, but not just at the cell surface. There's a lot of intracellular expression as well, and I think it's increasingly recognised that these receptors can be active, and the mislocalization of their signals is also probably really important in these cancer cells developing extra properties. So I think there's a lot of potential for targeted drug delivery sort of at the subcellular level as well in this condition.
So I guess that's a pretty good overview, I think. I learned a lot. Targeting and sensitivity, and a way to go with a lot of it, but very interesting. Another question? No, other way.
When developing small molecule therapies, the assessment of safety in a preclinical context is pretty well defined. You have to go through a series of steps. I would imagine that it's not well defined for nano at this point. And is somebody working on that?
Safety. A physician, I think. Andrew.
So the basic principles always apply. I think that actually, to some degree, nanotechnology has advantages over some of the other biologics which are currently being put into clinical studies at the moment, because one of the reasons that the testing is so rigorous and so complicated is because you're actually using a biologic molecule which in itself may have significant side effects. And to be able to purify them, to be able to test for them, can be quite problematic. So I think that it's not necessarily that difficult. And in fact, I'd almost go so far as to say there are precedents which suggest that nanotechnology may be a lot easier than biologics. There is, for example, a therapeutic which was developed in Australia, in Western Australia, called surspheres. Surspheres are essentially inorganic small particles, like silica particles, that has derivatized on the outside an isotope, yttrium-90. Okay? And these are actually administered via an injection into usually the artery which is supplying that region of the body that you're trying to treat the cancer in, usually in the liver. And so you'll have one of our radiology colleagues will do a catheterization into the hepatic artery. There's a lot of other tests to check that it's right for this particular patient. And then they inject these yttrium-labelled particles into the liver. Now, this particular technology was developed in Western Australia. It went through a series of clinical trials, not that many, and it was subsequently approved in the US in a very, very quick manner, with not as much necessarily safety data as you see with other biologics, because it was considered to be a device. And if it's a device, it's a completely different section of the FDA and a completely different criteria in terms of how it's approved. Yeah. And so, they were very lucky, and they've been able to actually get it approved in numerous countries around the world. So there are precedents for, nanotechnology, if you like. Of course, every type of technology will be assessed differently. But if it's an inorganic particle, or a very simple chemical particle, it actually isn't anywhere near as complicated potentially as if it's a biologic.
I wonder if you're using the same device in a sense to deliver a different drug. Does the previous test of the device with something else actually count as well, apart from the testing of the drug, which will have already been tested in another context?
So some aspects of the preclinical background and reassurance for the agencies or the ethics committees will be there, but they would expect there to be a replication of the same testing as was done previously.
Thanks.
So one of the papers that Peter put up there was from Mark Davis, who was visiting us from the US earlier this year, and he's had the approval for the testing of one of his nanoparticles for a particular therapy. And he said that it was remarkably easy to get his approval of these particles, but the trick for him was to show that these particles weren't degradable, because what the FDA wanted to be able to see was that they want to know that every possible degradation product of your material is going to be non-toxic. And so if you have a material that you can show that you put into the, well, they showed it in animals, that they put it into the animal and then it was excreted in exactly the same form, then it was actually a much simpler approval process, because they didn't have to look for potentially toxic degradation products, because they'd established that it wasn't actually degrading. And so this is an advantage that can potentially be for not just inorganic materials but for organic polymers. If you make a lot of the time we think we want to make them degrade in the body as quickly as possible, but sometimes if we can make something that we know can be excreted and excreted in a non-degraded form, then that's actually quite a good solution for the delivery.
I guess tattoos are kind of, well, they're not even nanoparticles that stick around, aren't they?
Yeah.
The other aspect is that if you're using the particular molecule itself as a delivery system, it intrinsically has a slightly easier regulatory pathway, as opposed to it being the actual drug itself.
Another question? I've sort of been wondering whether there are any potential technologies for getting stuff through the blood-brain barrier, because we have this problem in cancer particularly that you can have particularly an antibody therapeutic that will work well peripherally, but then you get problems with tumours in the brain because it just doesn't get through. Is there a nanotech strategy that anyone's thinking about?
So interestingly, I think that the evidence more recently has shown that certainly if you've got metastatic disease in the brain, or if you have high-grade primary brain tumours, there's an intrinsic low or high-level disruption of the blood-brain barrier such that it's not necessarily as great an impediment as people may thought. No. In fact, there's an antibody that we developed in our lab that we ended up licensing to Abbott that has a toxin on it which is now in clinical trials, and I've just come from a meeting in the US in Miami where we're seeing very, very impressive responses, and we've actually imaged the antibody and shown that where you have high-grade brain tumours as well as metastatic, you do tend to see on MR that characteristic gadolinium enhancement, which is one of the ways that they predict if it's particularly malignant, and then the antibody can often penetrate through. Interestingly, small molecules like tyrosine kinase inhibitors, although they're smaller, often have problems of penetrating the blood-brain barrier more than antibodies do. It's not necessarily intuitive, but that's often the case, and so nowadays there are efforts to design small molecules in different ways so that they can better penetrate the blood-brain barrier. At the end of the day, if you have a slight disruption of the blood-brain barrier, you will have fenestration gaps in that membrane which will allow any nanoparticle to get through. I think the key is for it to be able to not just get out of the initial blood-brain region, but also to penetrate through. And for that you need a more targeted strategy, and there are many ways you can do that.
Well, we've been thinking a bit about getting, circumventing the blood-brain barrier and taking a leaf out of cocaine addicts, because if you snort it, it'll go straight, I guess, to your brain. And so, and there has been studies to show that intranasal delivery, if you target the olfactory region, for this particular study showed, I think it was in Texas, showed that you can get exosomes up to the brain.
We used to think for a long time that one of the primary virus entry points was through the ethmoid plate into the brain. I'm not sure that's true, but it is more accessible. Another question?
So, Hannah, we're hearing a lot today about personalised medicine, particularly in cancer treatments and therapies. Is there a role for nanotechnology in personalised medicine?
I think definitely, right? So any of the personalized diagnostics, therapies, any of these things, but also, as I mentioned very briefly earlier today, I mean, we're actually part of a large consortium now in the UK that's funded called iSense, that involves several partners and also the London School of Tropical Medicine. And one of the things we've been doing there is developing these apps for iPhones and smartphones, so that you can essentially do the readout yourself on site, but of course it could be in the home as well, that sort of thing, and then have it all linked up actually with Public Health England and with the NHS, so that all these systems are automatically updated. And I think that's kind of ahead of the times, but I think it's the way that things are heading, and this is a very large consortium grant that we have on that at the moment. So yeah, nanotechnology within all these portable device type formats, but I think also linked up to telecoms. I mean, there's more people now that have, there's more mobile phones on the planet than people, which is kind of astounding, isn't it? I mean, just more phones actually than people at the moment. So being able to link all of that for diagnostics and treatment and more integrated healthcare, I think is really important.
Yeah, it's incredible. I mean, I was in was in Egypt and sort of driving through more or less the desert, and there's the Bedouin talking on their mobile phones, and so I don't think we know where any information goes, quite frankly. It goes everywhere.
Well, I guess there's a big driver for the point of care diagnostics market, and a lot of technologies are starting to get to the point where they're pretty much small enough, cheap enough, and to be rolled out. I think there's another implication as well. I mean, there are a few implications to this. I mean, for example, in point of care diagnostics, is whether industry would welcome that. For example, I mean, medical practitioners, in terms of how, if you would be able to do it yourself, screen yourself, what are the implications in terms of interpreting the results. There are also, on the other side, the companies which can do gene sequencing, for example. You can pretty much get your gene sequenced, but what implications there are in terms of, for example, if your insurance company gets hold of that. And so I think there's also the other side of things beyond the technology, the ethical side, which I'm not sure whether the issues have been worked out quite as yet.
I mean, the doctors already complain about Dr. Google. If it becomes Dr. iPhone as well, I think that might be really quite complicated.
Just building on that point of care diagnostics with self-analysis, I suppose two questions. One is the incidence of false positives better or worse in the area, or the same? But also, two, how do the panel globally work in the area envisage you deal with quite often, so when professionals issue out an issue of diagnosis, there's a psychological impact depending on the condition, what you've got, and having a human person in the interaction as opposed to a smartphone saying, you're going to die. How do you deal with it? Well, seriously, how do you deal with psychological fallout? What can be quite a serious diagnosis? And also, mesh over the fact that it could be a false positive.
Well, I think for diseases that are very serious, as you say, even if you have a point of care test, I mean, it's one that you should probably do with your physician, or at least do and get the result from your physician. And certainly for a lot of these very serious infectious diseases or early cancers and so on, then that's exactly what you would do. You wouldn't sit in your room at home and suddenly go, oh my God, I have a brain tumor. I mean, that would be a really bad way to get the news, right? So as far as I know, most of those possible diagnoses, the responsible way to do it, of course, is to get it through your physician. But being able to maybe do those on site at the GP's surgery or things like that, obviously, or within remote locations in the developing world, that's very helpful to be able to have that capability.
I guess I'll just make two comments. The one is that to be able to do tests at either in the home, for example, or at the GP surgery, I think we're interestingly approaching an environment where that will become both more possible but also more difficult. So what I mean by that is that I think the technology is leaping well ahead of our regulatory environment as to how we do that. You know, if you work in a medical area, the regulatory compliance that we face is incredible, and it's getting more and more. You know, everything from, you know, the simple blood tests that we used to do, you know, just run it through a standard machine and just get the result out. You now have to go through these ISO compliances, and the level of complexity for that is becoming higher and higher. And so that many small GP surgeries, for example, that used to be able to do small tests now are actually going the other way. They just don't. So it'll be interesting to see how that goes. Going back to the comment about Dr. Google, I always find that very interesting when I see patients as well, because one of the first things I typically ask them is well, what do you know about your condition and what questions do you have? And from that I can very easily work out just how much they've been on Google. And usually it's very easy to find out. And my comment to them often is that, you know, I often go on Google and I get confused, and I actually know a little bit about what we're talking about today. So a lot of people, I think, are very interested in getting as much information as they can. But I also think that people are, in general, very cognizant of the fact that there is just so much information out there. How do you sieve it? How do you know what's real or not? And that's where the interaction with your physician is obviously very important. But it's a challenge that we all face.
You know, I was at a medical legal meeting recently, and the doctors all complain about Dr. Google, and the judges all complain about the Googling juror, which is a real problem for them, because they're supposed to get the law from the judge. Yeah. I'd like to ask you a question. We've just had a rollout of a wonderful drug for treating hepatitis C virus infection. It's actually a curative drug for this terrible persistent infection, but the thing is selling for $80,000 plus a shot in the United States. Do you envisage that sort of issue with the nano approach? Are we going to get massively expensive products from various companies, or because we're often using technology that's already there in some senses, do you think we can roll this out and actually decrease some healthcare costs?
So, a very interesting question. I mean, when you look at some of the very expensive drugs, the justification for them is always just how long, how expensive, and how complicated it's been to develop them. And the big companies will always say, well, you know, we've had to do 2,000 patient studies, and it's cost us X hundreds of millions of dollars to develop the drug. And often that's true. But then it's also a matter of how much can they charge. And that's why in the US things cost more than they do elsewhere. I think the way it's going is that unless the regulatory environment changes and we can be smarter about how we develop drugs so that they can be done quicker, faster and cheaper, it's going to be difficult to have a substantial reduction in price. But the way a lot of the regulatory authorities are going is that they're insisting that for some of these very expensive drugs, you not only prove that it can work, but you have to now show in which patients it will work. And I think that's where nanomedicine can play a very important role, because accompanying diagnostics, accompanying predictive theranostics will become almost mandatory for these very expensive drugs, because they don't necessarily all work 100%. Hepatitis C might be, you know, getting closer to the exception, but there will be an increasing demand that before it's approved, you have to have that, or the regulatory authorities will say you have to have a predictive test and we'll only pay you if it actually does work.
Okay, so we've got 5 minutes left. I'm going to use my chairman's prerogative, and I'm going to ask each of the panelists to say where they think nanomedicine is going to be in 10 years' time. 1 minute.
So, I mean, from the technology point of view, I think, we've taken a leaf out of, for example, the silicon chip industry, where you can pretty much make these computer chips using photolithography, and you can make millions and billions of them, mass manufacture them, and Apple doesn't sell you the computer very cheap, but you can be rest assured, when they make those chips, it's probably a few dollars a pop. And one great example I've seen that transferred to pharmaceutical manufacturing, and back to Peter's question about whether you can actually use that to bring down the cost, is these huge arrays of nanopillars on which they just roll coat just a liquid on top, and because of the wetting just on the post, you can just coat and tablet these very, very quickly, very, very uniformly, of very specific size or whatever shape you want. And so that's an example where potentially, you can use nanotechnology in pharmaceutical manufacturing, and that this is actually a good manufacturing practice already.
I suppose from a basic biology perspective, I can tell you where I think there's some really huge potential, and where I would really like to see some of this go, is being able to target these drugs, not just to particular cells, but as I said before, to subcellular areas, because a lot of the problems with a lot of drugs at the moment is side effects, and not just off-target effects, but on-target effects, but in the wrong spot in the body. And as Nigel was presenting earlier today, that there's now evidence as well that receptors in the same cell have quite different functions, whether they're on the cell surface or whether they're inside the cell. So I think there's huge potential to be able to target these drugs really, really discreetly, and then hopefully eliminate a lot of the side effect profiles that we see.
So I think it can go in many, many ways, but one of the really exciting things I think is going to be this convergence of nano with other fields, so things like telecoms and other fields as well. But also I think the ability to much better control cell fate, so through things like much better cell transfection and that sort of thing. So being able to really target specific cells and control their biology, which is completely fundamental to all diseases, really.
So I think from a clinical medicine perspective, we already have examples of where diagnostics and therapeutics based on nanotechnology exist. I think in 10 years' time there'll be even more. But what I hope would be even more exciting is that we can not only have a platform, we can personalise it for each individual patient, and that would be wonderful.
And so I suppose coming back a bit to Molly's point of our ability to improve the delivery and the effectiveness of a lot of therapeutics, an example that was given to me a while ago is that a virus is 10 to 100,000 times more effective at getting its DNA into a cell than our absolute best nanotechnology system at the moment. And so we know that we have a huge potential to be able to tap the power of a virus, and we even think that the viruses may not even be that efficient in themselves. And so if we can understand how viruses do that, and if we can start to try and mimic that with our nanomaterials, then I think that opens up a lot of different therapies that, in terms of your cost, things like sRNA, very expensive to deliver. But if we can deliver 10,000 times less, then the price becomes more feasible as a proper cure rather than only people who can afford $80,000.
As a virologist, though, don't make any self-replicating nanoparticles.
Well, exactly, and that's why...
That should be the first law.
And
first law of nanomics. I mean, the first law of robotics: don't make a robot that kills people.
Well, and that's exactly the point, and that's where one of the advantages of the nanotechnology is that we know we've got something like a virus that is very good at delivering the DNA, but we don't particularly want to be treating people with the viruses because we know there's been cases where there's been the side effects of that. And so if we can make sure that...
We did actually treat some cancer patients quite early on, not we, the science, with really very dangerous viruses, thinking that they kill off the tumour cells. Fortunately, they didn't kill the patients. I think that's about where we'll leave it. I think we should thank our panel for their informative and interesting